So I'm excited to share with you some of the things that I've been doing. Most of the stuff that I do is on integrative and multi-scale modeling. This is a little bit more specialized, where I'll be talking about some gene regulatory network modeling that we're doing in sorghum bicolor, specifically in the stem. All right. So bioenergy crops, they hold a great deal of potential for a sustainable future. And that is because they are used as renewable biofuels. The plants themselves can be factories. So we call them plants as factories, where we can engineer bioproducts to accumulate in them to have some secondary uses. And they are also contributors and can make quite a significant contribution to climate mitigation through carbon capture and sequestration below ground. And so I always like to talk about crop idiotypes. And an idiotype is an ideal plant that can thrive under a given scenario. And so if we talk about an idiotype for a bioenergy crop, it would be one that would be high yielding, resilient to stress, and sustainably produced. And so I call it the poster child of bioenergy crops is bioenergy sorghum. And this is because it is naturally drought tolerant, highly nutrient use efficient, and it is also high yielding. There's a great deal of above ground biomass. And much of these traits can be attributed to sorghum's very large and deep rooting system, but also due to its stem. And I've got to be honest, I have not spent much of my career thinking about plant stems. I guess I've kind of always taken them for granted, right? They're the thing that holds the leaves, and they transport stuff between roots and shoots, which are the real stars of the show. But as it turns out, sorghum stems are actually pretty amazing. They can grow between 3 and 5 meters long, and they account for about 83% of the harvested biomass. Stem height has been shown to be an early indicator in breeding programs that are selecting for bioenergy sorghum genotypes with increased biomass yield. And if we go inside of the stem and look at the cells, so they're really here in blue is the pith parenchyma cells, and they act as carbon storage organs throughout plant growth. And of course, that means that they can send resources up and down the plant as needed. And of course, stems are the backbone of the plant. So what makes these stems so amazing really comes down to their cell walls. So, cell walls are unique to plants, and in specific cell types, plants even double down on their cell walls and synthesize these thick secondary cell walls. And so that's what this red arrow is pointing to here. These very thick looking things are the secondary cell walls around the vasculature. So these secondary cell walls, they give plants their mechanical strength to stand upright. They provide a layer of protection against pests and pathogens. And of course, they enable that transport of water and nutrients over long distances and under very high pressures. And an extreme example of this is water and nutrient, the long distance transport of that over 300 feet in the giant sequoia. And so I have to tell this story, is that my whole life, ever since I was a child, I wanted to see these giant trees that grow out in the West in California in the United States, but I've never managed to take a trip out there and go to Sequoia National Park in California. But then I come to Bonn, and lo and behold, I've seen several giant sequoias. And so thank you for helping this dream of mine come true. So I had to work that into the talk somewhere. So anyway, a lot of research has been carried out to learn about the structure and the biosynthesis of these secondary cell walls, really in the hopes of engineering their composition for specific purposes. So talking about bioenergy crops, the lignocellulosic biomass has been exploited for the production of liquid biofuels. So myself and some of my collaborators that are on a Department of Energy funded project, we really wanted to tap into the potential of sorghum stems for a few different purposes. One of our questions is that we're really interested in how different cell types within a stem are regulated, both temporally and spatially. So for example, you know, how is it that two cell types that are right next to each other can have such different functions? So we would like to uncover the regulatory differences between cells that make secondary cell walls and those that don't. Second, we are interested in the stem as a storage organ. I had already mentioned that the pith functions as a carbon storage, but we want to investigate if it would be possible or even a good place for to store other bioproducts that might be produced in the plant, such as fatty acids that can be extracted and easily converted to biofuels. So to learn more about STEM biology, we collaborated with a Department of Energy lab called EMSL. It's part of the Pacific Northwest National Laboratory. And then I have collaborators at Texas A&M University. And then this is Jia Fu. He's a graduate student in my lab, and all of the analyses that I'm going to present to you, he did. He's a great student. All right. So what we did with EMSL is we performed laser capture microdissection, and we did this to isolate the cell types and collect them for RNA-seq analysis. And so the figure here on the left shows pre-cut and post-cut using the laser to isolate those cell types. All right, so then after the RNA-seq, the first thing that we did was perform a clustering analysis of the derived data, and this showed us a few things that we were very pleased to see. Of course, that the replicates are clustering together, and then that different cell types form distinct clusters. So for example, up here in the right, upper right corner, we have the pith parenchyma, epidermal, and in this figure, I'm showing subepidermal cell types that I won't continue to talk about. But anyway, these cluster together, and they cluster in this space very far apart from the three different vascular cell types that we isolated, which includes the phloem, xylem sclerenchyma, and xylem parenchyma. So next we wanted to pull out the genes that are uniquely expressed in each cell type. We did this using an equation called the TAW index. What you need to know about that is that genes that have a TAW of 1 are only expressed in one cell type, whereas those that have a TAW of 0 are expressed in every cell type. So we used a TAW cutoff of greater than or equal to 0.8 to get our list of genes that are predominantly expressed in a single cell type. So then to check the validity of our cutting and our analysis, we searched these gene lists for known marker genes for each cell type. And of course, we were pleased to find many of the known marker genes for these cell types in our sorghum samples. So in the epidermis, there's one called lipid transfer protein 1. Altered phloem development is found in the phloem, and xylem nacdomain is in the xylem cells. Up until now, there has not been a specific marker gene for pith parenchyma cells, but we did find a study in the Arabidopsis inflorescens stem that they had identified several pith-expressed genes. We did an intersection of our pith-specific genes with theirs and found an overlap of 66 pith-expressed genes from Arabidopsis. As another sanity check, we determined if the cell type specific genes were involved in common biological processes that we would expect to find in these different cell types. And indeed, we found highly enriched gene ontology terms for each group of genes. So these are summaries of biological processes. So for example, if we look in the epidermis, we find wax and fatty acid metabolic processes, shared processes between the epidermis and xylem sclerenchyma for cell wall biosynthesis and organization. In the phloem and xylem parenchyma, we find overrepresented Go terms for various transport processes, which is what they do, and in the pith, photosynthesis processes. So again, these were all things that we were hoping to see, and then we have some other biological processes that are upregulated in these cell types that will need further investigation. That could be some novel things. Okay, so the cluster plot that I showed earlier told us that there was this clear separation between the vascular cell types and the non-vascular cell types. And so we wondered, could we use our laser capture microdissection samples, these transcriptomes, if we could use the data to find the genes and the biological processes that are driving this separation. So what Gia came up with is that he first performed pairwise comparisons between all combinations of vascular and non-vascular cell types. And he did this to get a list of differentially expressed genes. So specifically what we're looking at here in the left are all of the upregulated genes across the vascular cell types when compared to the non-vascular cell types. And if we take all of these gene lists and intersect them to find what is common among them, commonly upregulated genes, we found 127 commonly upregulated genes. Gia did a gene ontology analysis on those and found that all of these transport processes, this is almost 100% different types of transport processes. Okay, so that's what's common among the vascular cell types. And then on the other side of the coin, we look to see what was common among the non-vascular cell types, epidermal and pith cells. These are the genes that are upregulated in non-vascular, which by virtue means that they're downregulated in vascular cell types. Okay, so again, if we take the intersection of these genes, we found 54 in common between pith and epidermal cell types. We did Go term analysis on that and found an overrepresentation of processes for photosynthesis and light response. And so this suggests that the upregulation of transport processes distinguishes vascular cell types from non-vascular cell types. And those non-vascular types are really distinguished by the upregulation of genes involved in photosynthesis. So these seem to be the functional processes that are separating these transcriptomes. Okay. Another question that we had was whether the cell type specific genes are commonly regulated or not. And we used a bioinformatic approach to examine gene promoters, searching for cis-regulatory elements that can be bound by transcription factors, which can turn on the expression of those genes. And what we found was that each cell type specific gene set has significantly enriched motifs. And in this heat map, you can only read it vertically and not horizontally. And so if we look, for example, at the epidermis cell types, we found that 95% of the epidermal specific genes had an enrichment of BZIP transcription factor family binding sites in their promoter region. The pith parenchyma cells, 95% of them have C2H2 binding sites. And then if we look at the vascular cell types, we found that the majority of them have GATA or G2-like transcription factor family binding sites in their promoters. And so the important or takeaway message here is really that this suggests that distinct transcription factor families are playing a role in regulating the cell type specific expression of these genes. Okay. So next, now we did a little bit of modeling. We used this promoter binding information to build gene regulatory network models. And what they do is that they predict interactions among genes. And since it's regulatory, we're really interested in regulatory interactions between transcription factors and their potential target genes. So since we're interested in cell type specific expression and what's going on there, we first only built gene regulatory networks that included only cell type specific genes and cell type specific transcription factors. And we found that the cell type specific transcription factors only have regulatory edges with about 40 to 50% of the genes depending on the cell type. So we're missing a lot of the regulatory landscape. So to capture more of it, we had to expand our gene regulatory networks to include non-cell type specific transcription factors. So this means that these are genes that are highly expressed in these LCM samples, but they have a low tau value. Okay, but they're still expressed, and so of course they can still have a regulatory function within that cell type. When we expanded the gene regulatory network, we found that the non-cell type specific transcription factors have regulatory edges with 65 to 98% of the cell type specific genes, again, depending on the cell type. But what was interesting, what I found interesting, was that these non-cell type specific transcription factors are predicted to be regulated by cell type specific transcription factors. So you can think of it as a hierarchy, and that's what I'm attempting to demonstrate here in this figure, is this hierarchical regulatory pathway of these cell type specific transcription factors regulate the non-cell type specific transcription factors, which in turn regulate a lot of the cell type specific genes. So the takeaway here is that these observations imply that there are two modes of regulation occurring within these cell types. There's both direct and indirect modes of regulation by cell type specific transcription factors to turn on the spatially explicit expression of genes. All right, so now I'm going to summarize this first part. We used a laser capture microdissection to enable the identification of genes that are uniquely expressed in each of 5 distinct cell types. We found that the differentiation between vascular and non-vascular cell types is driven by functional differences in biological processes, including transport processes that are upregulated in vascular cell types, while photosynthetic processes are upregulated in non-vascular cell types. Sorry, it just got really loud outside. We also found that there are suites of cell type specific genes that share common cis-regulatory elements in their putative promoter regions. And we found that there are 2 modes of regulation for cell type specific gene expression, both direct and indirect, by these cell type specific transcription factors. So now I'm going to move on to the second part of the story. And the second part is that we really wanted to use this laser capture microdissection data to gain new insights into the well-studied process of secondary cell wall formation in stems, which is not so well studied in bioenergy sorghum. And so this analysis was led by Brian McKinley, who is part of John Mullett's group at Texas A&M University, and these folks are experts in bioenergy sorghum, so very good collaborators on this project. Okay, so Brian first used sorghum stem developmental data from a previous study that he and John had done to identify genes involved in secondary cell wall biosynthesis in sorghum. So this developmental data revealed that these secondary cell walls form as vegetative internodes mature. So in this figure here on the right, this is a picture of the internodes of bioenergy sorghum about 60 days after planting. And I've lined up the images of the scanning images of these cell types from youngest to oldest. So youngest is this internode 1, oldest is internode 4. And so what can be seen in this figure is that these internodes 1 through 3, if we zero in on these vascular bundles, they're blue. And this is because they're stained with Alcian blue, which indicates primary cell walls. But as the vegetative internode matures, the vascular bundles then get stained red with this safranin O, which indicates accumulation of lignin-containing secondary cell walls. Okay, so then also using this developmental data set, Brian looked at the expression of some marker genes for secondary cell wall biosynthesis. And these are called cellulose synthase genes, CESA, and there are 3 different ones, 4, 7, and 9, that he looked at their expression over development and found again that as the internodes mature, you get induction of expression of these genes. All right, so since they're well-known marker genes, Brian used these to anchor very targeted gene regulatory network analysis. So again, this is just using the developmental data. He's wanting to pull out what are all of the genes in sorghum that are involved in secondary cell wall formation. So he found that there were about 1,200 genes that are co-expressed with the cellulose synthase genes, and this table is just a subset of that network, and this highlights the other known genes that are involved in secondary cell wall formation. And so that includes the cellulose synthase genes and some others, genes that are involved in xylem formation, including GACs, TBLs, FLAs, and then genes that are involved in lignin biosynthesis. So it's basically they found their old friends in their network, which was a confidence building thing. So the next thing that Brian did was that he identified the transcription factors that are predicted to interact with the promoters of these genes. And fortunately, this also includes many well-known transcription factors that are involved in secondary cell wall formation, also some new ones that were identified in sorghum. And so here, these blue boxes indicate transcription factor binding to the secondary cell wall gene promoters. And I can draw your attention just to one group here. This is a group of MIB transcription factors, and they are more central to lignin pathway expression. And this is something that was also consistent with prior research and other species that found this regulatory relationship between MIB transcription factors and genes involved in lignin biosynthesis. So now Brian used our spatial data, so the laser capture microdissection data, to reveal spatial variation in the regulation of secondary cell wall biosynthesis during internode growth. And so again, this is just a subset of the results, but even what this shows is that most of these genes that are involved in secondary cell wall formation are highly expressed in xylem sclerenchyma cells, also known as xylem fiber cells expressed at intermediate levels in the epidermis and expressed at very low levels or not at all in the other cell types, including pith, phloem, and xylem parenchyma. Okay, and I had shown you before that lignin is a good marker for secondary cell wall accumulation over internode differentiation. So Brian used cross sections of the sorghum stems and stained them with this safranin O, which stains lignin red. And so here we can see that the lignin is really accumulating on cell walls of cells surrounding the vascular bundles, mostly the xylem fiber cells, and on epidermis cell walls, and not in pith and phloem cells. So this data, this staining data, is in agreement with the secondary cell wall gene regulatory network, showing that there's higher expression of these genes and lignin in xylem, sclerenchyma, and epidermal cell types. I had mentioned that many of the transcription factors in our network are known regulators of secondary cell wall formation in other species. And in fact, this is a diagram of the canonical secondary cell wall gene regulatory network that was first identified in Arabidopsis, and additional information has been added for some grasses. And I've highlighted here the transcription factors that were also found in our network, and you can see that there are several. So while our data is consistent with the expectation for the overall pathway, our spatial transcriptomes tell us exactly where these important regulators are expressed. So for example, SNDs, so up here at the top of the network, these are considered master regulators of secondary cell wall formation. So the SND genes, the homologs in sorghum, is called SND2, and they're very highly expressed in the xylem sclerenchyma cells and not expressed in the other cell types, whereas the other master regulators, the NST genes, are expressed in both the epidermis and the xylem sclerenchyma cells. And so this could potentially explain why secondary cell wall gene expression is highest in xylem sclerenchyma, moderate in the epidermis, and very low or not on in the other cell types. Okay, so in summary for this part, we performed a targeted gene regulatory network analysis, or I should say Brian did, and found that secondary cell wall formation in sorghum is similar to what has been established in other species. That seems maybe not super exciting, but it's just one of those basic science things that it hadn't been established in sorghum, which is the organism that we're working with. So that was important for us. And then we found that our cell type specific transcriptomes were able to provide spatial resolution of which secondary cell wall genes are expressed in which cell types and which transcription factors contribute to the regulatory differences in secondary cell wall formation in xylem sclerenchyma versus the epidermis. So we have a lot more work to do. This is very new data that I was happy to share with you all. So our next steps, we want to follow up on some of the specific relationships that we identified in our secondary cell wall analysis and get some more molecular support for those predictions. And then we have complementary data that we collected at the reproductive stage. I may not have mentioned this, but everything that I was showing you was at a vegetative stage. And so by comparing those data sets, we will then be able to learn about temporal differences in spatial regulation across these cell types. And then at present, we're still working with EMSL and collecting and analyzing spatial proteomics and metabolomics data, which we think will enhance our network analysis. And I guess that's it. That's a lot of work. Okay, but none of this would be possible without our collaborators. I mentioned that Gia is a wonderful graduate student working with me who did all of the cell type specific analysis. John and Brian are at Texas A&M and focused on the secondary cell wall. Jasmine also is at Texas and collected all the samples. On some of the other future direction projects, we're working with Conchita Swaminathan and Brandon James at Hudson Alpha. And then here's a list of our partners at EMSL, and they all were, Galia Orr is the leader of that group at EMSL. Funding for this project is from all angles of the Department of Energy. We have 2 different BRCs, which are the big centers that are funded by DOE. CABI is the one that I'm part of, and John and Brian are part of the Great Lakes GL BRC. And then, of course, EMSL is funded by DOE. Okay, I made it at 30 minutes. All right, so that's it. Thank you.